So this is how that comes in. So these guys are found in the synaptic cleft. Again, that's the space between the two membranes of the nerve and the muscle. So that's how that comes in. So again, if you're releasing the signal through acetylcholine, that is the neurotransmitter, that's the signal chemical from the nerve, then these are cholinergic synapses. The other ones that we saw here are the nerves that release the signal as adrenaline. For example, or norepinephrine, or even epinephrine. These are the adrenergic synapses. And what part of the autonomic nervous system uses more epinephrine or norepinephrine? The sympathetic, the fight or flight. The acetylcholine is used by the parasympathetic. That is part of the autonomic nervous system. This will coincide with chapter 14. Okay? So you have these groups of nerves from the autonomic nervous system that you have no control, and if it's releasing acetylcholine, it's the parasympathetic. In your handout, you have that on page 3. Okay? This is from chapter 14, right here. Okay, on chapter 14, this is where it has this box right here. Remember, these are front and back, page 3. All right, so that box has the parasympathetic and the sympathetic flight or fight response. The parasympathetic is called the rest and response. It used to be called the rest and digest, so no longer is that. This only focuses on digestion. Okay, so this is utilized extensively by the sympathetic nervous system. We're going to see that network of autonomic nerves later on. So we have right here norepinephrine. Again, it's part of the autonomic nervous system used by the sympathetic nervous system, so that's part of that relation. You also see this neurotransmitter, norepinephrine, used in the central nervous system. So in the brain and spinal cord, between those nerves, you could also use this type of neurotransmitter, norepinephrine. And of course, the adrenaline, the one we actually use that common name as adrenaline, the epinephrine is used by the autonomic nervous system, and usually this one is associated with the nerves activating the endocrine system. So these are glands, part of the secretion of glands that don't have ducts. To stimulate them, you will use epinephrine. Again, this is part of the sympathetic nervous system as well, to activate them. So we're going to see how these work in the central nervous system. They're going to help you to produce the onset of this circadian rhythm of sleeping and being awake. So that's important. And we know that when you have adrenaline or the norepinephrine, that's going to make you more alert. See that? It's going to make you more alert because that's going to keep the function going of those cycles. But again, this could also coincide in some other areas of the central nervous system that could also end up also producing an inhibitory action. But those are very rare. Usually they excite. They're usually exciting. Okay? So that is the type of nerves that release adrenaline or norepinephrine, or the adrenergic synapses. And then we have four other hormones. That's dopamine, serotonin, GABA. Remember the dopamine, we saw that with the Muhammad Ali and Michael J. Fox. Again, with dopamine, when you don't have enough of the dopamine, you're not regulating the contraction of the muscle. And the result is the muscle is contracting spontaneously. And as a result, you don't have this coordinated movement. And that's what deals with this result of what we call the problem here, what they have with Parkinson's. See that Parkinson's disease. Low dopamine. So that was, again, the result of not having the dopamine to inhibit these areas. When you look at the serotonin, again, this is from the amino acid tryptophan. So this is, again, another neurotransmitter, very important. This hormone is, the main component is tryptophan, and tryptophan is linked with the ability for you to fall asleep. Like when you eat a lot of what? Turkey. Yeah, we usually blame it on the turkey, but keep in mind that chicken has more tryptophan. than the turkey. What's the reason why the turkey got blamed? Because you overate during that time. But it has very little compared to the chicken, so you had to eat a lot. See that? Even the leftovers, again. So to boost it up. So tryptophan is not just found in the turkey, it's actually less than the chicken. The chicken has a lot. Also beef has a lot, okay? More than the turkey. Anyways, so the tryptophan, again, it is a signal, and serotonin and dopamine in certain regions of the brain, especially in the brain, is going to shut off these excitatory nerves that want to send the impulse, the action potential. You got to shut them down so that you could regulate the cycle of sleeping, so you could go to sleep. So that is, again, an interesting aspect of that. And then we have this very famous one, GABA. This GABA is very unique because, once again, it actually terminates the signal at a very unique place of the neuron. Not in the dendrites or cell body. It actually terminates, annihilates the signal at the synaptic knob. And that is why this is called, that location where you release GABA, again, is called the oxaloxonic synapse, where the synaptic terminal connects to another synaptic terminal at that point. That's the oxaloxonic synapse. Okay? So briefly, what you see here is that indirectly it's going to block the entry of calcium so that you don't produce that neurotransmitter. So that boils down to that. So again, these are major neurotransmitters you should be familiar with, including the other two, acetylcholine and epinephrine. Okay? You should know them. But these are very important because they help to down-regulate. Remember, the other ones are to excite. These are going to shut down. So usually that's why these are important. Shutting down nerve impulses. Okay? All right. So these are just additional information. I'm not going to go into them. But let's look at another important aspect that we need to talk about. Let me see if I still have it, because we're looking at the end of this chapter. I want to go back to the excitatory nerves and the inhibitory nerves just to wrap it up. Gotta go into the old one. Just to give you a quick summary of the action here with the nerves. All right. So one of the uniqueness that I just want to re-emphasize is that most of the nerves that are regulating other excitatory nerves are these that are blocking the axon hillock. And this is basically what you're going to see. Inhibitory nerves are going to be guarding this trigger zone, the axon hillock. So basically, inhibitory nerves are going to be around this area, and excitatory nerves are going to bind furthest away from the trigger zone. So usually, these nerves that are blocking this area make it very difficult to excite the nerve that you see here. So the inhibitory nerves is the one that you have to overcome with the stimulus. And they're really usually located around the trigger zone, around the axon hillock. The excitatory nerves intervention is usually at the dendrites or the cell body, away from the trigger zone. Remember that the trigger zone was important because this is where you have the highest concentration of voltage-radiated channels. The signal that you get from other excitatory nerves will be at the dendrites or cell body, trying to get to the axon hillock. Remember, that signal is called graded potential. If it's strong enough, then it will overcome the inhibitory, and then it becomes an action potential. Okay? But remember, this is how you regulate the complexity of nerves. Not all nerves you can easily stimulate. Some of these complex nerves you have to overcome the inhibitory nerves. Okay? So that is why Let me see if I can find a picture. I just wanted to show you the difference between the excitatory nerves, and this is the only thing I have, I don't know why. But it's basically abbreviated excitatory nerves, excitatory post-synaptic potential, and inhibitory nerves, the IPSP and the EPSP. I don't know why I cannot find those slides. That's the last thing I want to make sure you understand. How did I put them in there? Oh yeah, let me go over this also to wrap up chapter 6 before I terminate. We move into the next one. Saltatory or continuous. The saltatory conduction means that you're not going to be activating the entire membrane of the axon. It's only the areas where there is no insulation, no myelination. So remember, a saltatory conduction means that the signal is going to be skipping from node to node, every node of Ravier. So remember that the signal is faster in a saltatory conduction because of the myelination. Versus the continuous means there is no myelination, so you have to go through the entire membrane. So it's going to take longer for you to open and close voltage rated channels. Again, this continuous or saltatory is referring to the axon region, how fast the signal passes through that. All right? So that was an important point that I wanted to re-emphasize. Any questions on those two? So that's only on the axon. If it's myelinated, it's saltatory. It skips. Faster signal. You send it from the axon hillock to the synaptic terminal very quickly. If it's no myelination, the signal is a little bit slower. We're talking milliseconds slower, right? It's like saying, did you see the difference? Okay. Well, anyways, if you didn't, it's just milliseconds. All right. There we go. So here is how you're going to be able to overcome the inhibitory nerves by temporal or spatial summation. So this is an important point that you need to know. So remember, the inhibitory are blocking at the trigger zone, right? So therefore, to make sure you overcome the inhibition of these inhibitory neurons that are blocking the graded potential signal to activate the axon hillock, you have to send the signal over and over. This is the trickle effect, the temporal summation. It will add on over time, and then it'll excite the nerve. Or spatial summation means that two nerves send the signal at the same time to excite the nerve, to help it overcome the inhibition. So that is how these two come into play when you're looking at exciting the nerve. So the nerve is not simply excited by sending the signal over to it. You have to subsequently send it over and over, temporal summation, or have more than one nerve send the signal. That's going to help you. These two ways is how you're going to be able to overcome the inhibition, the action of the inhibitory nerves. The inhibitory nerves, called the IPSP, inhibitory postsynaptic potential, is produced by nerves that want to stop the signal. And they do it by making the membrane more negative. Instead of by being minus 70, they make it minus 90. So therefore, the signal coming in from the nerve, that plus 15 millivolts, will not be enough. You need to add more of this significant stimulus. That's what this is referring to. So this will be the last thing I want to talk about for this chapter. The physiology of how you produce that action potential. Questions here? So remember, why do we need these two? Why do we need them? This type of action. If you want to excite the nerve, this is what you need. Why do you need these two? And the answer is because the inhibitory nerves, they're making the membrane more negative. They're regulating the nerve not to fire up. So the inhibitory neurons make the membrane more negative. So to solve the issue, the nerve has to keep on resending the signal. What is that called?
The temporal?
Temporal summation. Or you could send it from two nerves. Spatial summation. And that's going to be able to solve the issue of the membrane being too negative. See that? So it's like a boost. Either way, by these two reactions, that's how you're going to be overcoming the inhibitory neurons. What region of the neuron is the inhibitory neuron attaching to?
The trigger.
Yeah, the trigger zone, the axon hillock, around that region. Remember, the correct name is axon hillock, not trigger zone. Okay? It's the axon hillock. We're assimilating it to a trigger zone. The correct name is the axon hillock. So that's how you're going to overcome it. All right. Don't forget, you will be required to know about the action potential. What is depolarization? What is the movement of the ion? What's the name of the ion? What part of the graph is depolarization?
Oh my God.
What is repolarization in the action potential graph? I'm going to ask you to draw the graph. Get that. That way I don't ask you no questions. I'm just going to draw the graph and explain it. Okay? So again, you should be familiar with this graph. Yeah, you like me now, huh? Again, this was taking you step by step. You see that? So I gave you this right here. This diagram. See that? I'm explaining it here. All right, so again, the membrane is negative. Why? Well, because it is not open. These voltage radio channels are not open. It's negative because of the leak channels that produce that negative charge to be higher inside the cell than outside, more positive. And the membrane is negative because of the phospholipid and protein. See that? And of course, that produces that resting state. You want to activate that membrane of the nerve. This is referring to the axon. Then, of course, you've got to have a significant amount of stimulus. To reach this point right here is what we call threshold. What's the significant stimulus amount that you need? Plus 15. That is enough to depolarize the membrane to a point that is going to open up these voltage radio channels. So 70 minus 70 plus 15 gives you minus 55. So that's how these will open. That's reaching threshold. That's what we call reaching threshold. This first part of the graph is depolarization. What is the name of the ion? Sodium. What is it doing? What is its movement? Into the cell. That's right. Well, it opens and they go into the neural cell. That's why it makes it more positive to the highest amount, which is plus 30. And then they close up. What's the next thing that happens when these voltage radio channels for sodium close up? The potassium opens. But remember, the potassium is
Inside the cell.
yeah, remember, inside, but coming out. So you're losing them. So that's why it goes back to negative. So this is what we call repolarization, because the potassium is moving out. See that? What do you have? What do you call this area right here? What does that mean? That's right. It goes below the minus 70. So it goes to minus, being more negative, minus 90. Why? So potassium are still a few open. So it makes it more negative. So that's the idea that we see here with the graph. You should be able to label the y-axis. We're going to call this. Yeah. See that?
So
Or millivolts, MV.
So
And this time.
the repolarization, is it from 3 to 4?
Yes. It's the downhill.
Then from 4 to 5 is the hypo.
Yeah. Well, this specific region right here, hypopolarization. Really, it's just 5. Okay? So that's, again, something I will ask you. So now we're moving into the brain. So now we're going to be talking about the brain. So let's go ahead and talk about the brain. And this is chapter 12. So with chapter 12, what we have here is the handout. The handout is what I'm going to hold you accountable for, all right? So this handout right here on the first page, if you look at it, this green handout, so this green handout, all right. So this green handout is the one that has the information for chapter 12, 13, and 14. You're not going to be tested on chapter 15. So your test that's coming up is over just chapter 11, 12, 13, and 14. Yeah, why not 10? Because it's a sliding filament theory and rigor mortis, that's it. That's why it's not a big deal. All right? But it's 15 points. 10 points for the sliding filament theory, 5 points for the rigor mortis. Remember, if you want more points, you need to add on to the rigor mortis. I'm sorry, to the sliding filament theory. So again, your test basically is going to have this also. All right? The majority is going to be fill in the blank, is going to be from this handout. I'm going to ask you, if you look at this handout, I'm going to ask you, which is the one that controls, appetite and temperature in the brain? Hypothalamus. See that? So which one is the one that has the ability for memory? You're looking at the conscious thought process. You see that? That's the cerebrum. See that? What about visual reflex, auditory reflex? Which is the visual reflex? That is on the midbrain. The second box. See that? Superior canaliculi. Which is the auditory reflex? Inferior canaliculi. Yeah, second box. Okay. So remember, the first box is the forebrain. That whole thing up here. See that? And then the second box is the midbrain. The third box is the hindbrain. Okay, those are common names. We got to know their official names. So that's what we're going to be looking at now. All right? As you can see here, this network of the brain, we're still discussing what really is the brain. We still don't know. Nobody can answer yet. How do you make memories? Again, the thought, collection. How does it stay? How does it happen? We still don't know. See what I mean? We just know that it happens, but we don't know how memory is kept. It's very difficult to understand. The brain has also been called a hologram. It's just a projection of whatever these neurons are doing. They're firing up, they're projecting it. And they're getting the information, the input from the environment again, projecting it now to a substantial way that you're sensing it, sensing the environment. Because really, there's nothing there. But now you're converting it to a form. See, that's the quantum physics. Anyways. So let's look at this area right here. I want to focus on the areas of the diencephalon. The diencephalon includes this area right in the center. So this is the thalamus and hypothalamus. So I want you to make a bracket. So what is another name for the thalamus and hypothalamus? Diencephalon. Diencephalon. Okay? And you can see this is part of the top of the brainstem. And then we see the brain. The brain is very unique. You see all these crevices that it has? I don't want no crevices on there. Or do you want these folds? Yes. And yes, you do want it. This is what we call the, there's a name for this. It just left my mind. The sulci. Yeah, but the folding. There's a specific name that refers to the intestine-like structures that you see here. Yeah, they're sulci. See that? These are the folds that you see here. But the formation of these folds, it is important that you need to produce them. And you produce them very early on, all the way to age 2. That's when the brain stops growing, the cerebrum. Well, just the overall site. But what you don't see that is still growing is what we call the limbic system. What is the limbic system in charge of? You look at it here. The last one in the first box.
Emotional behavior.
Yes, emotional behavior. A lot of emotions right there. Okay? All right, we also have the amygdala that we're going to be seeing. Anyways, so this membrane folding is important. You need to make these folds. Okay? The plasticity of the brain is important. So the best way to explain these folds is what I saw with Star Trek. Okay, you have point A over here and point B. What's the fastest way to go from point A to point B? See that? That's what your synapses do. They shorten the distance. Instead of making a neuron connected, they fold on itself, and that enhances your ability to ride a bike, to walk, to do anything. Anything that you've been doing very good at, that's where you have the best synapse. If you're good at eating, we know you are. You're good at eating because we see you. Anyways, the point is that you make these synapses. You have good coordination. But it's repetitive action that you need to do. And of course, this area that we talked about is right in the center. Again, this is like an egg-shaped or bean-shaped structure, the thalamus. Process, integrates, relays. So this is all the control center. Everything of the sensing information that you are picking up from your nerves, your peripheral nerves, from your sensors, receptors in your skin, to your blood, and also your blood vessels. All of that sensory information is going to go up to the thalamus, this area of the diencephalon. So it's the ascending nerves that is part of the sensory. Descending, or what we call the efferent nerves, going away, are part of the actions to do a movement. See that? So when you sense stuff, you're going to bring it up to the thalamus, and that's why here, this area, the diencephalon, which includes the thalamus, is going to be able to send it to the right location. So that's the big picture. Everything is sent to that area, and we'll send it to this perspective zone. In the brain, you have this segment right here. See this right here, right? This line right in the center. All right? That is a shallow indentation. That's called a sulcus. That is the central sulcus. What side of the brain is this? What side are we looking at? What side? Left side. Why is that important? Because the left side, you have that sulcus, as well as the central sulcus, as well as the folds, the gyri. Very important. So I'm going to always draw you this picture. You got to be familiar with it. This is a simple picture of the brain. Kind of flatten out, but anyways. I'm going to do that. That's what you're going to see on your lab test 4. I'm going to ask you, what is that? What is this fold in the front called? What is this fold in the back? The gyri. See that? That is on this handout. On your handy dandy handout. That is on page 2. Look at the top box. Has pre-central, the primary motor cortex, and the primary sensory cortex. Pre-central gyrus and post-central gyrus. So guess what's the pre? The fold over here. What's the post? The fold over here. See that? And in between, the central sulcus. So we need to know what a gyra is. The gyra is a fold. Sulcus is a shallow indentation. Fissure, deeper separation. So those are points that you need to be able to understand from this chapter. All right? So once again, as we look at the brain and you do a cross section on it, you're going to see that there's holes on it. These holes right here are the ventricles. We're going to be looking at that. And if you do the same thing to the spinal cord, once again, you have this matter on the outside. What's the matter called? White. And what is this? The gray. White outside, gray inside. Look over here. See the gray and white on the inside. Gray is the outer crust. It's inverted. What I told you before. This gives you the butterfly, the wings that you see here on the spinal cord. See that? Again, you see the gray matter, white matter in the center. Okay? All right. So this is just telling you the developmental stages. Again, how this brain comes about. You see, it basically is just like a long tube right there, and it basically bulges out. See that? And it starts to produce these regions that we've been talking about. We also see the mesencephalon. The mesencephalon region, again, that location is below the diencephalon. So if we go back to this picture that we had at the beginning, okay? So the mesencephalon is right here. That's your, according to this handout, that's your midbrain. So I want you to put that name for the midbrain, mesencephalon. So draw a bracket over the entire midbrain. So what's the entire midbrain? Superior, inferior, red, nuclei, substantia nigra, all the way to reticular, in this case, formation, or RAS, R-A-S. It's also called that now. Reticular formation is the same thing as RAS. All of that. The midbrain is mesencephalon. Mesencephalon. So the entire midbrain, the specific name is mesencephalon, not midbrain. All right? So once again, you have the central sulcus. So this is on the second page, top box. You see that you have the central sulcus that is going to separate the pre-fold from the post-fold. The precentral gyrus, what does that contain? That contains the gray matter, the cortex, for what? For motor. See that? Precentral gyrus, motor. What does that motor refer to? Movement. Yeah, any movement in the body, any muscle you're going to move in the body, that area contains it. In other words, if you touch this area of the brain, you'll be making that person move. Jerk those areas. Move the hand, move the leg. As we go upper to this area, you'll be on the face, the tongue. So all this area has been already identified, mapped out. Because when you have open brain surgery, the brain has no pain sensors. So you can touch it. The person doesn't, just like you saw with Hannibal. That's the only thing that's true. You could open it, there's no pain. But that's why they have to wake you up. So if you touch it, you're going to be moving those areas. But this one, post-central gyrus, what does that do? Sensory. And as you look at this is primary sensory cortex. All right? The precentral gyrus. So that's important for us to know. So this is the feeling. So if you touch anywhere on this side of this gyrus, this fold, the gyrus is the fold, post-central gyrus, then the person's going to feel something on their body, all through their body, depending on where you're touching. Okay? The higher up you go in here, towards the middle, that's on the face and tongue. Okay? You're going to see that because they mapped it out pretty good in our textbook. They have it pretty mapped out. So let's look at the lobes of the brain. We have the frontal lobe, because it coincides with the frontal bone. Parietal lobe, see that? Temporal, occipital. All of these are able to do a specific action. We're going to see that. In the left side, basically around this lateral fissure, see right here, this lobe and the frontal lobe are really separated. But as you go towards the parietal, this lateral fissure becomes a sulcus, shallow indentation. So they're not really very much separation. When you go further out, they are very separate. Just like you see in this line right here that totally separates left and right side of the brain. This longitudinal fissure. Okay, the longitudinal fissure separates left and right brain. It's a deep indentation, just like the lateral fissure. But this lateral fissure goes, it's almost like going towards the beach. You start hitting land, and then it gets, less deep, less deep. So that's what's going on here. So it becomes a sulcus. So that's what you're going to see: lateral fissure, lateral sulcus. Right? But at the edges, right here, that's a fissure. Okay? That's what they found out with Einstein, that he had it different. More gray matter in that area. Okay. All right. The insula, as you can see here, is between the separation of those two areas. The lateral fissure, you open it up, and deep to that, you have the insula. So that's another region of the brain. Okay. Remember that when it comes to humans, we do have the largest brain. How many pounds is our brain of an adult human? the weight. It's in pounds. 3 pounds. Females weight 2. See, that's why they have less brain than males, right? It's true. The weight says it. No, of course it's not. Who has the highest IQ? A female. That's right. She's in the more than Einstein, and she's still alive. We have a lot of women that have very high IQs, including one of these politicians. Anyways, so the point here, once again, making a big issue of these areas. And then what I'm interested is right here. Look at this, Broca's area. I'm gonna make a big issue. It's also on your page 2, Broca's area, regions of the brain. You see it right there, Broca's area. But it's not only the Broca's, you have the taste, you have the olfactory, you have the auditory, the visual. You should know where these are located because it's corresponding to this. But what's unique about the Broca's area? What does it say? Control speech.
You have a stroke there, you can't talk.
That's right. But there's been evidence to demonstrate that this is not always the case. If you lose this area, what the brain has done is that it does compensate for other regions. This is something we can't explain how it does that. Why did it do it on this person and not in another person with a stroke? See that? So again, there are different areas of the brain, but this is not set in stone that only here. And a good example of that was the NBC reporter. Remember that was with shrapnel in Iraq? The entire brain, his left side, actually, it was the left side was knocked out. He doesn't have anything. And then within 6 months, he was speaking, talking. He knew his history. He knew how to do his job. And he didn't move as well, but he still was able to recover without, that part of the brain. I mean, it's amazing. How come people that just get a hit there, then that's it, they're dying? I mean, how come this person didn't die? Or the person that got that arrow through him? Or of course, the most common one, the most popular one was in the railroad. Remember where they were blasting, and they shot one of the metal pipes through him, through his skull, and that's where they saw that. Hey, well, that's kind of weird. It went through the frontal lobe. It went like this. Yeah. Oh yeah, he lost his wife, he became a drunk, but yeah, it changed him completely. But then in the 1940s, we had another person, I think I still have the picture, that was curing seizures by destroying this through your eyes. And he was praised as being like a god, like amazing. He's curing all these people. No, he was making them more dumbfounded. So again, that was a technique that was basically praised, and now we know that it was making them worse. It was taking away the seizures, taking away their erratic behavior, but they were just now
catatonic.
Yeah. So now they could sit there forever.
They got a lobotomy.
Yeah. Have you seen it? I'm going to show you those pictures. So that's been the problems with these medical advancements, because that was a big thing. He would take little kids, like, yeah, come into my office, and then come out, and the kid was like a robot. But he cured them. So anyways, so they started to investigate this more. That's what he was doing. He was destroying the frontal lobe. The frontal lobe is also a problem with crime. When it comes to people ending up in jail, we're going to see a lot of problems here. Especially males have a problem that this doesn't fully develop. See that? We're going to talk about that. Why is it that as a parent, you have the right to make decisions for your kid up to the age of 21? Because they're still going to be stuck on stupid. So all your dumb decisions come about because this is not, you think it was a good idea to get off the car and sit on the top of the roof of it while it was still driving. Stuff like that. Like, surfing. It's not good. Anyways, so the frontal lobe not fully developed is a consequence. That's why you shouldn't drink at all, because you are affecting it. So that's why we have the loss up to age 21. That's when that develops. Anyway, so we'll keep on talking about this. Don't forget, this is chapters 12, 13, and 14. All right, so your test that's coming up for a lecture is dealing with that. This afternoon, remember, we're going to continue on with the video, the autopsy video. So it's very important you be here. We're going to make an emphasis, again, for your test next week. Practical. It'll be over chapter pages 196, 197, right? So the majority is superficial muscles. Yes?
This video is the one that's gonna show us the
Deep muscles. Some of the deep, not all of them. Some of the deep muscles, yes. Those are the ones you should be familiar with. All right, we'll see you at 1.
Yeah, please be fed. You mean like the last one? I don't have a book. I know, nobody wants to bring me this. No, I have the last one. I don't have a book. I need the book. I look, I brought the lab manual and
Yes. There was some,
I believe